The purpose of this series is to explore how the bacterial flagellum evolved, and to answer the questions put forth by Destin from Smarter Every Day about the process of evolution. How does evolution build supposedly irreducibly complex structures, sophisticated molecular machines like the bacterial flagellum? Flagella are the little structures that bacteria use to swim, and when we're zoomed out like this, these flagella are not super impressive to look at, but zoomed in under an electron microscope, we start to see how beautiful this structure is. Here is a close-up of the base of a flagellum, and here we are looking at a highly accurate computer model showing us how the flagellum works. It is a natural, incredibly efficient, ion-powered microscopic rotary motor made of protein. The structure is so spectacular that if you go on TikTok, you'll find people claiming there that it was invented by aliens. This, of course, is not in line with our best scientific understanding, but I do understand the awe that structures like this inspire. Recently, my colleague, my fellow YouTuber, Destin, from Smarter Every Day, he put out a request for someone who understands biology to explain to him how on earth did something this complex evolve. It's a very sophisticated structure, and it's sitting right there in bacteria. This is the Stated Clearly Darwin Day special, How Did Bacterial Flagella Evolve? Episode 1, What Do We Know and How Do We Know It? Normally, here on the Stated Clearly channel, I do animations about genetics and evolution and chemistry, but today is Darwin Day. It's kind of become a tradition on this channel for me on Darwin's birthday to do a little lesson about evolution. I don't do it every year, but this year, instead of doing a single lesson, I'm going to do a series of lessons because this question that Destin has, how did bacterial flagella evolve? This is a question that I do not think should be taken lightly. Most people in my audience are probably too young to remember this, but when Destin and I were in high school, back in the 90s and then after high school, so in the early 2000s, this question, how did bacterial flagella evolve, was heavily debated in the public. Several groups of people worried that evolution was in conflict with Christianity. Not all Christians believe this, of course. Lots of people have found ways to accept the science and also maintain their faith. But there are pockets of Christians, even still today, who remain uncomfortable with evolutionary biology. In the 90s and in the early 2000s, tensions boiled over into heated debates. These fights were not happening on your grandma's Facebook page. These debates were happening on the news. In the evening, you would turn on your TV and you would see panels of people arguing about the evolution of bacterial flagella on C-SPAN, the way people currently argue about Donald Trump. I was still a student back then. I wasn't yet teaching biology, but it would have been a crazy time to be a biology teacher. It was insane. But the questions these people had about evolution were actually really interesting. Science teachers who really knew their stuff enjoyed the challenge. And the questions are fairly similar to the questions that Destin has today.
People say, well, how can something this complex come to be out of nothing? The logic goes like this. If this motor system is composed of complex individual parts, and all these parts work together to perform the overall function of rotating, then how did the individual parts come to be? Did it all have to happen at the same time? Or is there some evolutionary advantage to the cell for every intermediate stage of development? Is 15% of this motor advantageous to the cell? What function would 50% of the structure perform? What were the steps these components took to assemble into such a complex molecular machine in the first place?
These are really fun questions to think about. Whether you know it or not, Destin, you have reworded, you've recapitulated what is often called the argument against evolution from irreducible complexity. The most famous version of this argument comes from Michael Behe. He was the guy that we saw on the news back in the early 2000s. He coined the term irreducible complexity, but he did not invent that argument. His doubt about evolution's abilities is a very common doubt for many people who are new to the idea of evolution. The oldest example of that argument that I was able to find dates back to the mid-1800s. It's in a book by Victorian theologian Robert Mackenzie Beverley. Biology teachers have been responding to concerns similar to this, concerns about irreducible complexity, almost constantly ever since the 1800s. Your questions, Destin, they're not difficult to answer. It really only takes a few sentences per question. The problem is, quick responses like I just gave here, unless you have a really good understanding of how evolution works, and unless you have a really good understanding of how proteins function, these responses will not feel satisfying. It's going to seem like I'm just doing some sort of a hand-wavy evolution of the gaps type argument. That's not what I'm doing, but it might seem like that if you're not familiar with these topics. So, Destin, out of respect for you and out of respect for my audience, here I have prepared a series of lessons, the series of lessons that we should have had back in the 90s. This is going to be a crash course on flagella evolution, starting today with how scientists study it, what we know and how we know it. Then there will be several lessons on how evolution works. Evolution by natural selection is a natural engineering process that does not require an actual engineer. And this is not a baseless assertion. I'm going to show you actual observations in the wild, observations that lead us to this conclusion, and direct observations in the lab of evolution happening right before our very eyes. We know that this process works because we have seen the process work. After the basics, we're going to do a deep dive into the evolutionary processes that you normally never learn about in high school or even in college unless you're majoring in biology. These are things like co-option, duplication, and recruitment. Following this, we're going to learn about molecular machines, also known as protein complexes. Biology is filled with spectacular machines. The human hand itself, this is a spectacular machine. There's pulleys and levers inside there. There's little sensors and grip pads on the skin. I think the reason that people have never really made arguments against evolution based on the human hand, I think the reason for that is, well, first of all, it's probably because scientists and the general public already have a pretty good idea about how hands evolved, right? All you have to do is look around a bit and you realize that human hands are modified mammal paws, and mammal paws are modified amphibian, blah, 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 all the way back to fish. But I think the main reason people have not spent a lot of time arguing against evolution based on the human hand is because we're not that impressed with our hands. We're used to seeing them. We take them for granted. Meanwhile, the bacterial flagellum is a rotary motor with perfectly symmetrical rings and fibers. We're not really used to seeing perfect symmetry in nature. In fact, if someone happens to have a perfectly symmetrical face, a lot of times they'll get paid millions of dollars by Hollywood to make movies. But symmetry at the molecular level is actually very common. You see it in ice crystals. You see it everywhere. Symmetrical rings and rods they're some of the easiest structures to produce at the level of protein complexes. They're so easy, in fact, that they can actually become a problem. Sickle cell anemia, the blood disease, it's caused by a mutation that triggers hemoglobin to form beautifully symmetrical but horribly dangerous rods inside your blood cells. Depending on the mutations, they sometimes form this really interesting double-barreled shotgun structure. It's a structure that people have thought about using to produce man-made molecular machines. Protein is not just a nutrient found in beans and meat. Proteins are amazing molecules. They can be rigid and brittle. They can be soft and bendy. That flexibility, along with their chemical charges, means that proteins behave sort of like squishy magnetic Legos. Magnetic Legos that diffuse freely, unbothered by gravity, through the liquid inside the cell. Evolution gets to play with squishy magnetic Legos in zero gravity. This is awesome. In that lesson, we learn how proteins fold, and we learn how protein complexes self-assemble. And I could just show, pictures from textbooks or, 3D graphics and so on. But what I've actually done is this here is a little, it's a little clay polymer model that I made. It's got magnets in it. And these magnets represent the charges that proteins have on their surfaces. And this is an actual model of a real protein complex that actually exists in the bacterial flagellum. This is the MOT-A complex. We're going to learn all about it, and I'm going to do a little experiment showing how self-assembly works. I'm very excited about that. This series will end with a detailed look at our best current model on the evolution of bacterial flagella, and that model can be found in this paper right here, if you want to do a little reading ahead. So, today's lesson is how do scientists study bacterial flagella, the evolution of bacterial flagella. What do we know and how do we know it? The motto of science is nullius in verba, which means take no one's word. Science is all about observation, backing up any claims you make with observation. So let's make some scientific observations together ourselves. Here in this little drop of water, there are thousands of bacteria. In fact, there are probably millions of bacteria. This is a fairly cloudy sample. Bacteria are so incredibly small that millions could easily be fitting in the single drop. One of the problems with that is that they're too small to see well under a microscope like this. This is a light microscope, and the wavelength of light is so big that it's hard to see details in bacteria. The really nice images that you see, those are always electron microscope images or something else. But that's okay. I have a trick. Here I have a little glass slip that I'm going to put over the sample. What I'm going to do is I'm going to wait a couple of minutes because the bacteria in the middle, those that like oxygen, they're quickly going to run out of oxygen. And those who like oxygen and have flagella, not all bacteria have flagella, they are going to start swimming and they're going to go towards the edge of that little slip that I put on here. We will be able to see them really clearly as a swarm around the oxygenated places on this slide. It's going to be really cool, but we got to wait a minute. So let's go back up to the office. Okay, I washed my hands very carefully, and I also added some extra drops of water to that sample because I don't want the bacteria to dry out while I'm up here talking. I've got some important stuff to cover here, and I don't want to rush myself. So they will be fine. Evolutionary biologists have three main projects they work on. First, they directly observe how evolution works today. That can be done by observing organisms in the wild over really long periods of time, or it can be done faster by comparing different populations of the same species in different environments. It can also be done through the study of mutations and new traits that pop up in domestic plants and animals, or through careful lab experiments. Even though evolution is a very slow process, it happens step by step, and it happens over many, many generations. Evolution can be studied and measured in real time if we're looking at organisms that reproduce fast, that have short lifespans. These would be things like bacteria and fruit flies. Those are very common organisms for evolutionary biologists to study in the lab. And here in a little bit, you and I will go back downstairs to study them ourselves. We're going to get our own little taste of project number 1 right here. The second thing that evolutionary biologists do is they collect data showing what organisms were like in the past. This is where things like fossil hunting come in. The past can also be studied indirectly through comparative anatomy and comparative genomics. DNA sequences give us tons of information about the past, indirectly, but tons of information. And now there is a new trick in town often called protein resurrection, which I'm going to talk about a lot later on in this series. The third thing that evolutionary biologists do, they take all of this data they've collected and they use it for the construction of evolutionary models. So the first two projects here, those are observational science. People are observing things in the wild, observing things in the lab, observing things in our genomes. That last activity is sometimes called historical science. It works like forensic science. You collect all the data you can about a topic today, and then use that data to reconstruct the past. In forensic science, you reconstruct a crime scene, right? So what is an example of an evolutionary model? Well, you probably heard of an evolutionary tree. These are evolutionary models showing which species are most closely related to other species. We also have stepwise evolutionary models. These show the possible or the most likely pathways a particular trait took as it was evolving. This, Destin, is what you are after. You want a step-by-step evolutionary model for the bacterial flagellum. You want to know what it looked like at different stages of its evolution, and you want to know how the cell was using it during each one of those stages. Don't worry, these models do exist. There's a really nice one in this paper right here, and we will go over it in detail in a future lesson. We're going to save that for a couple weeks from now. Just to recap, because this is very important, evolutionary biologists learn how evolution works by directly observing evolution today and by collecting data from the past. They then use that data, that knowledge, they use what they know from direct observation to build evolutionary models. These evolutionary models are hypotheses. They are hypotheses about what happened in the past. And a lot of people, they hear that word hypothesis and they say, oh, So what you're telling me is that scientists don't know anything. They don't know what happened in the past. Well, yeah, we can't know with absolute certainty what happened in the past, but there is a lot that we can know. Even though we don't know with certainty every detail about how bacterial flagella evolved, we do know, because of direct observations both in the wild and in the lab, we know that the normal process of evolution by natural selection has all the tools necessary to construct irreducibly complex systems through a gradual, step-by-step process. In the next 2 lessons, next week and the week after, we're going to go over the evidence for this in detail, and it is fascinating evidence. It really is cool what we're going to learn in the next couple of weeks. So again, here is what we know. We know that the normal process of evolution by natural selection has all the tools needed to build these so-called irreducibly complex systems. This includes systems like the bacterial flagellum. Scientists have also produced evolutionary models. These are hypotheses, showing how that process of evolution may have actually played out step by step. All right, let's go back downstairs and check out those bacteria. Let's go make some scientific observations for ourselves. Okay, let's see what we've got here. I'm hoping that I didn't wait too long and they're all dead. That would suck. This is cool. We don't have just bacteria here. We also have some predators of bacteria, some protists. So protists are eukaryotic cells. They're a lot more closely related to animals. They're not animals, they're single-celled organisms, but they're way bigger than bacteria. Let's check out the bacteria first. That cloud, that line, that cloud, that is the bacteria. They are swimming very rapidly with their little flagella. Look how efficient those little motors are. It's amazing. There's way too many bacteria here to count. And then the bigger guys are protists. They're probably, they're some sort of paramecium. Okay. Remember, I said that paramecium hunt bacteria. One of them just hit payday there. It's really neat that we're seeing the bacteria and the protist here together, because protists also have things that are sometimes called flagella. In paramecium, we call them cilia. There are lots of little hairs all around the organism. Here's a better shot of one where you can see all that detail. A lot of people think that those are flagella. They are not flagella. They're not flagella like the bacterial flagella is a flagella. This here is another protist. It's another large single-celled organism. Instead of lots of little cilia, it has one giant cilium. Confusingly, scientists call this a flagellum. It's not structured at all like the bacterial flagellum, but they call it a flagellum. And this is the same type of flagellum that sperm cells have. It's very, very similar. It's evolutionarily related to the flagellum that sperm cells have. The process of evolution is a mindless tinkering process, and because of this, it reinvents the wheel over and over again. Each time it rediscovers the wheel, it pushes and pulls and modifies that wheel into a huge diversity of forms. Flagella-like structures have evolved at least 3 times independently. It evolved once in bacteria and then diversified into all the forms we see in bacteria. It evolved again differently in archaea. And then in eukaryotes, we have structures that are not true rotary motors. Instead, they are whips that look sort of like rotary motors. People often think that all of these structures are the same thing. They are not the same thing. They have ended up performing the same function, but they are very different at the molecular level. Evolution is a tinkering designer, not a rational designer. So that's one common misconception, but an even more important misconception is the idea that cells have to move in order to survive. They don't. Moving is very helpful, but it's not essential. A lot of times the anti-evolution people will tell me that they don't believe that flagella could evolve because before flagella evolved, bacteria wouldn't have flagella to swim, and bacteria need flagella in order to survive. That's not the case. Just like how plants don't need to run in order to survive, lots of microorganisms don't need to move in order to survive. Here I'm showing you yeast. That's not bacteria, but it's big, so we can see it easily. This species of yeast, and then lots of species of bacteria, they do not have any way to move, and they survive just fine. This is an African beer yeast that's eating the sugar in the beer, and it's reproducing. This is sped up, of course. Flagella, the ability to move, came along later in life. So the first living cells, we don't know how they came to be, but they did not need all the fancy equipment that modern bacteria have. Bacteria themselves have evolved lots of different ways to move, not just flagella. You also have gliding motility, where bacteria attach to a surface and then kind of move their skin around. There's also a really neat thing that bacteria do. They have little fibers that stick out of their body. Those fibers are sticky. It can easily grow them and pull them back in. They will use those like grappling hooks and pull themselves along a surface. That's what we call twitching motility. And all of that is just in bacteria. The ability to move around, even though it's not essential, it's so important that any organism that develops some sort of a way to move, even just a little bit, that often gives it a survival advantage over those around it. The first cells on Earth did not need to move around because they didn't have any enemies that could move around. But now that lots of organisms can move around, it's a huge, huge advantage. And look at that motility. These guys can really swim fast. It's amazing what these little flagella can do. Whew, that was a lot of information, right? A lot of the details that we learned, especially down there, looking at the bacteria, a lot of those details you don't need to memorize to understand this stuff. Just a lot of that was to give you a taste of what types of information evolutionary biologists use when they're building their evolutionary models. It's a very careful process. A lot of data is collected. I'm going to stop things here today so you can digest all of that. But we are only just beginning. New episodes in this series will come out every week, every Thursday evening to be exact. So you know the drill. Like, subscribe, and hit that bell icon so that you don't miss the next episode. Also, share this video with all of your friends and everyone you know. Pester people on Facebook and Twitter and all those places. I've accidentally turned Darwin Day into Darwin Month. My apologies to the king. This episode was paid for by patrons over on patreon.com/stated clearly. And by the way, I hate to be that guy, but contributions are actually really important now for Stated Clearly. You might have noticed that over the past year, I have not produced many animations. And a big part of the reason for that is that there just hasn't been funding for it. I used to get a lot of my funding from grants, and those were grants given to scientists. Scientists would do research, and then they would give me a chunk of that grant money for outreach. But you might have noticed that in the last year, there's been a lot of drama and funding cuts in the U.S. surrounding science. So, yeah. Your personal contributions are very, very much appreciated right now. If you want to offer more than what would really make sense to donate through Patreon, Patreon takes a cut of the money. You can contact me through email, John, Jon, @statedclearly.com. There's also a contact form on my website, statedclearly.com. And then finally, huge shout out to Professor Morgan Beebe. His lab does extensive research on bacterial flagella, and this guy has spent hours with me making sure that I understand all of this information well enough to teach it to the world. Thank you, Dr. Beebe. You are a true gentleman, a scholar, a saint. Seriously, thank you for your help on this. Destin, I will see you next week.